Welcome everybody to the MGH brain-PBM clinic rounds. My name is Paolo Cassano. Today is May 19, 2023. And it is my great pleasure to introduce Dr. Denis Bragin, Associate Professor of Translational Neuroscience at Lovelace Biomedical and Department of Neurology at the University of New Mexico Medical School. He's going to talk today about photobiomodulation and the glymphatic system. Denis, please take it away. If you want to start sharing.
So, good morning, everybody. I would like to thank the organizer for the opportunity to give a presentation of the MGH PBM research series, and I also to acknowledge my collaborators at Saratov State University in Russia, New York Medical College, MGH, and ProNeurolite. Let me move it a little bit. Okay. So my topic is mainly based on our recent review publication on photobiomodulation and brain drainage system in the International Journal of Molecular Sciences. I'll also describe some of the seminal research on the use of photobiomodulation in brain drainage system activation. These studies were done for the first time by a group of Russian scientists at Saratov State University, in which I am proud to participate. This is here you can see some of the members of this large group, headed by Oksana Semyachkina-Gushkovskaya. So I'll start with an introduction in quite recently discovered, or better say, rediscovered brain glymphatic and meningeal lymphatic drainage systems. Afterwards, I will talk about photobiomodulation therapy in relation to the drainage system, involvement of nitric oxide, neuroprotection, and the ways of photobiomodulation therapy delivery. As we know, transcranial photobiomodulation is application of low levels of red or near-infrared light to stimulate tissue. Photobiomodulation has been demonstrated to be an effective approach for promoting cellular proliferation, microcirculation, and for relieving pain, edema, and various traumatic, acute, and chronic disease. It has been also shown to improve brain hemodynamics along with an increase in cerebral oxygenation and metabolic capacity. The increasing bodies of evidence suggest that photobiomodulation can ameliorate neuronal oxidative stress, neuroinflammation, and apoptosis while promoting neurogenesis and synaptogenesis. To date, no adverse effects have been reported for photobiomodulation. However, attention must be considered with higher power lasers due to hazard for retinal damage. The glymphatic system is a glial-dependent waste clearance pathway in the brain that drains away waste metabolic products and soluble proteins such as amyloid beta, for example, from brain parenchyma. The meningeal lymphatic is a network of lymphatic vessels located in the dura near the venous sinuses and meningeal arteries, responsible for draining immune cells, blood, small molecules, and excess fluid from the brain into deep cervical lymph nodes and further down to the lymphatic system. An impaired brain damage system can increase the incidence of neurovascular, neuroinflammatory, and neurodegenerative disease. Photobiomodulation therapy potentially can serve as a non-invasive neuroprotective therapy for maintaining and optimizing effective brain clearance. Recent preclinical research in rodents proving the efficiency of photobiomodulation for improving or restoring brain drainage system in several models of brain disease. As I already said, the glymphatic system is a recently discovered waste clearance system. It was discovered in 2012 by a group of researchers from University of Rochester, headed by Mike and Nedergaard. What they do, they used in vivo 2-photon imaging of small fluorescent tracer injected into the brain and found that CSF enters the mouse brain rapidly along the perivascular spaces of arterioles, and weight exchange with interstitial fluid. After that is cleared from the brain parenchyma via perivascular spaces surrounding large draining veins. The name Glymphatic system was given in recognition of its dependence on glial cells and the similarity of its function to the lymphatic system. On this animation, you can see the flow of CSF, ISF mix, the involvement of astrocytes, and the release of waste through the perivascular space of the veins. It should be noted that first who discovered brain waste removal via CSF was German surgeon Quincke in 19th century. He injected cinnabar into the subarachnoid space of free-moving animals and observed that dye was transported with CSF. However, it should be noted that he haven't seen that cinnabar entering the parenchyma, probably due to extremely large size of the molecules. So this scheme represents how does the glymphatic system works. This fluid transport of the glymphatic system can be divided into 4 distinct segments. First, cerebrospinal fluid is produced mainly by choroid plexus. Then arterial wall pulsatility drives CSF deep into the brain along perivascular spaces. CSF enters brain parenchyma within the neuropil, where interstitial fluid mixes with CSF, and this exchange is supported by aquaporine channels in astrocytes and feeds along the arterioles walls. And the fourth component is the glymphatic efflux, which consists of drainage of the fluid into the perivenous space. Astrocytic aquaporine water channels facilitate this perivenous efflux of the interstitial and CSF, which drains to the dural meningeal lymphatic vessels that can be called the fifth component and the final downstream clearance of the brain drainage system. Here it's just a more detailed picture of this processes. You see astrocytes along the vessel walls, and the aquaporine is involved in this fluid movement. So let's move on and discuss what is the meningeal lymphatic system. In general, the lymphatic system is a part of the immune system, complementary to the circulatory system, and it consists of a large network of lymphatic vessels and lymph nodes that move lymph throughout the body. And the main role of the lymphatic system is to act as a filter against microbes, organic waste, toxin, and other debris. Before 2015, it was believed that the brain does not have the lymphatic system, as it depicted on the image from the anatomy textbook.
I think we need to advance that slide.
I don't know what.
Brain drainage system.
I don't. Okay. So this is a brain without lymphatic system and lymphatic vessels with lymphatic nodes. However, in 2015, investigators from Virginia, headed by Jonathan Kipnis, discovered the meningeal lymphatic system in mice that activates during anesthesia using a specific lymphatic vessels dyes. You can see the fluorescent meningeal lymphatic vessels on this in vivo to photon microscopy images. In other experiments, they injected dye into the cisterna magna, and they found that dye was transported to the cervical lymph nodes. And this is how they shown the meningeal lymphatic function. At the same year, the group of Finnish investigators headed by Kari Alitalo, they developed transgenic mice with fluorescent lymphatic vessels, and they were able to absorb the lymphatic vessels in the meninges, and they also injected dye into the cisterna magna and found that the fluorescence appears in this deep cervical nodes after some times. Again, it should be noted that in 1787, an Italian physician, Paolo Mascagni, created a complete atlas of lymphatic system. And in addition to the mapping of the body lymphatic system, he depicted this vessel in meninges around the brain. To do that, he injected a mercury into the cisterna magna into about 1,000 corpses, and on 7 of them, he absorbed some vessels that were not blood vessels, and he saw that it's a lymphatic system. But since he absorbed it only in 7 corpses out of 1,000, the Society of Scientists dismissed the idea, and therefore we can say that meningeal lymphatic system was rediscovered almost quarter century after the first observation. So here is an updated images of picture of lymphatic system, including the meningeal lymphatic system and the brain. So the meningeal lymphatic vessels play a crucial role in maintaining brain homeostasis by draining macromolecules via cerebral spinal and interstitial fluid from the central nervous system to the cervical lymph nodes. And the CSF-ISF mix from the glymphatic system moves to the meningeal lymphatics. So these two systems work synergistically. And of course, impaired cerebral lymphatic system is considered as a risk factor for inflammatory diseases, neurovascular disease, and impaired recovery from brain injuries. As depicted here, even if glymphatic system is functional, when meningeal lymphatic system is impaired, the waste products cannot leave the brain and accumulates in the brain, even with active glymphatic system. Another part of the brainage system is a perineural space surrounding olfactory nerve and the cribriform plate. The cribriform plate is a fenestrated bony plate that separates cranial and nasal cavities and considered another key extracranial site of CSF outflow. CSF passes through cribriform plate along olfactory nerves to nasal lymphatics and enters cervical nerves, cervical lymph nodes. In other words, this is another pathway by which CSF is removed from the skull. So on this image, you can see the outflow of CSF through the cribriform plate. So here is a summarized scheme of how does the brain drainage system work, including lymphatics, meningeal lymphatics, and cribriform plate pathways. CSF through choroid plexus and ventricles moves to parenchyma through the glymphatic system, and then it moves to the periveneal system to the lymphatic vessels in the meninges and drains to the cervical lymph nodes. However, so far it was not clear how does the fluid with waste moves from the perivenous space to the lymphatic vessels in the meninges, because these two layers are separate. This year, this question was answered. The group of investigators found a fourth meningeal layer where the glymphatic and meningeal lymphatic system do connect. The traditional view is that the brain is surrounded by three layers: the dura, arachnoid, and pia mater. Montgard et al. Found a fourth meningeal layer and called it subarachnoid dynamic-like membrane, or SLIM. SLIM is immunophenotypically distinct from other meningeal layers and represent a tight subdividing layer in the subarachnoid space into two different compartments. It is the host of a large population of myeloid cells that increase in response to inflammation and aging. So this layer represents the innate immune niche, ideally to surveil the CSF. Functionally, the close opposition of SLIM with endothelial lining in the meningeal venous sinus allows direct exchange between CSF and venous blood and meningeal lymphatics. It has been already shown that several conditions can modulate brain clearance and influence the risk of developing neurodegenerative disease. It's a lifestyle, in particular sedentary, obesity, circadian disturbance, sleep quality, aging stress, and substance abuse, genetics, and pathology, hypertension, small vessel disease, TBI, cardiac disease, cardiovascular disorder, and apnea. An impaired brain drainage system is a risk for neurodegenerative, neuroinflammatory, and neurovascular disease, and tumors as impaired recovery from the brain injury cause waste proteins accumulation and aggregation, and this is a kind of a vicious cycle. This is just another picture showing more like cartoon accumulation of the waste due to sleep disturbances, stress, hypertension, and high levels of alcohol. It has been shown that brain drainage is modulated and driven by cardiac cycle, which drives vessel pulsatility, respirations that change intrathoracic pressure and affects brain ventricles pulsatility, neuronal activity that affects extracellular space volume, and vasodynamics. It has been also shown that brain drainage system is mostly active during the deep sleep phase and is suppressed in awake state. Exact cause of this suppressed brain drainage in awake state is unclear, but it's believed that it correlates with reduced CNS noradrenergic drive and increased CSF production during the sleep. On this MRI images in human, you can see the meningeal lymphatic vessels, which were already shown in humans. And on this cartoon, we see the CSF pulsation in the fourth ventricle as an example of a fluid drive. Okay, well, it doesn't work. Okay, let's talk now how does photobiomodulation can be related to brain drainage, sorry. Starting in 2018, a group of researchers from Russia has made efforts to test the possible modulatory effect of photobiomodulation on the brain drainage function. They started with testing the hypothesis that photobiomodulation promoted relaxation of lymphatic vessels might be one of the mechanisms for increasing permeability of lymphatic endothelium, thereby allowing larger molecules to be transported through the lymphatic vessels. Indeed, they found that in testing photobiomodulation increases diameter of lymphatic vessels in systole and diastole. In other series of experiments, they have been shown that photobiomodulation also activates meningeal lymphatics in the brain. Here you can see that after photobiomodulation, the fluorescent dye injected to the cisterna magna clears much faster than without photobiomodulation into the cervical lymph nodes. And it was concluded that this action is most likely a result of relaxation of lymphatic vessels via vasodilation process. In another experiment, it was used mouse model of intracranial hemorrhage. The autologous blood was injected into the brain, and after several sessions of photobiomodulation, it has been shown a much faster clearance of the blood from the brain. And you can see different meningeal lymphatic vessels at different regions, and the increase of the diameter of those vessels after the photobiomodulation. Same thing was with the injection of Evans blue. You see much faster accumulation of Evans blue in lymphatic cervical lymph nodes, fluorescence, than without photostimulation. The next question to be answered was the mechanism of dilatation of meningeal lymphatic vessels. The first candidate was nitric oxide. Nitric oxide is a well-known vasodilatator that plays a significant role in the homeostatic regulation of blood vessels and blood flow. Nitric oxide mainly produces vasodilatation by endothelial nitric oxide synthase pathway. It is suggested that photobiomodulation increases nitric oxide photoviability, sorry. Drink a little bit. Get dry. There are several mechanisms. First, it photodissociates nitric oxide from cytochrome C oxidase in mitochondria. Second, it increases nitric oxide bioavailability from intracellular stores, especially engaging heme proteins. And the third, it has been shown to increase nitric oxide production via nitric oxide synthase, increase expression level or phosphorylation. So using the specific inhibitor of nitric oxide, L-name, it has been shown that photobiomodulation causes vasodilatation in meningeal lymphatic vessels via nitric oxide synthase-dependent mechanisms. Here you see the animals without photostimulation, animals photostimulation with increased arterial diameters, and no change in meningeal lymphatic vessels, even with photobiomodulation. The follow-up studies revealed that photobiomodulation-mediated increase in the blood-brain permeability can result in further activation of lymphatic clearance of beta amyloid from the brain in the model of Alzheimer's disease, and that this process is most active during the sleep phase. So in this experiments, animal was monitored for the sleep phases, and it automatically triggered photobiomodulation at different phases. And as you see here, so this is another type of scheme of this experiment. And after 11 to 19 days of photostimulation, the animals were subjected to behavioral testing and histopathology. And you can see here accumulation of Alzheimer's plaques in naive animals without photobiomodulation. It was significantly less in animals that with photobiomodulation in awake state, and much less plaques in animals that were photobiomodulated during this deep phase of sleep. And this is a representative images, intact animal, no photostimulation during awake state, and photostimulation during sleep, much less plaques. And it's also shown much better improvement in neurobehavioral testing. This is just another image of the same experiment with accumulation of the beta amyloid beta in cervical lymph nodes. You see much more accumulation during the sleep phase during photostimulation. Here is another example of experiment. So the nasal brain lymphatic system through ethmoid bone is discussed as pathway for brain drug delivery overcoming the blood-brain barrier. However, in most cases, this method is not sufficient to achieve a therapeutic effect due to brain drug delivery in the short distance. In this study, it was demonstrated intranasal delivery of liposomes to the mouse brain, reaching glioblastoma. And this experiments with ablation of meningeal lymphatic network, an important role of meningeal pathway of intranasal delivery of liposomes to the brain has been demonstrated. So this data revealed that photostimulation can increase the delivery to the brain of liposomes. So this is a glioblastoma in the mice, and the liposomes delivered directly to glioblastoma through stimulation of the ethmoid bone. So with photobiomodulation therapy, from this we can not only increase the brain drainage, but we can deliver drug through the nasal cavity to the target organ. So as a result of these studies, oh, seems I missed some. I don't know, where is it? Disappeared. Okay. Anyway, so as a result of this study, several ways of photobiomodulation strategy to the brain has been recommended. It includes transcranial, intranasal, intraoral, intraoral, and intravascular approaches. Moreover, it has been suggested that photobiomodulation therapy targeted at remote tissues can produce indirect effects, providing neuroprotection to the brain via systemic mechanisms. And also, the best defined time for photobiomodulation drainage system is a deep sleep phase. From that, the patient should be monitored at night with some physiological monitoring device that will trigger photobiomodulation at deep sleep phase. And here is a PBM photobiomodulation scheme that is suggested to activate different parts of the brain drainage system. So the top part will activate meningeal lymphatics, the head band will activate glymphatic system, and the neck band will activate deep cervical node moving of the waste downstream to the lymphatic system. here is a suggested paths by proneural light. And another important aspect is intranasal photobiomodulation that has been shown to increase CBF, decrease blood viscosity, increase hemerology, and increase blood coagulopathy status. And of course, it improve lymphatic clearance through the ethmoid bone and cribriform plate. So with respect to the brain drainage system, intranasal PBM might also serve as an effective treatment of obstruction of the cribriform lymphatic route, thereby improving CSF outflow. Here is another paradigm how to do it, and modulation of cerebral autoregulation and reduction of inflammation. And in conclusion, I would like to say that brain drainage system plays an important role in maintaining the water and ion balance of the ISF and CSF, waste clearance, and reabsorption of micromolecular solids. And second, physiological function includes communication with the immune system, modulating immune surveillance and response of the brain. And photobiomodulation of the cranial and extracranial lymphatics may be a promising approach for the treatment of brain disorders. Thank you for your attention.
Thank you so much, Denis. This is quite a talk again, very, very fascinating. If you want to take your slide down, we can kind of open the discussion here. And yeah, anyone who wants to jump in, I have a few questions, but I hear that there are many people here. Yeah, also feel free to jump in. I don't see all of you at once, so I might not see if you raise your hand. Well, I guess I can start with a question here. So I was wondering if the effect that you have noticed related to photobiomodulation during sleep state are fairly local to where the light was shed, or are generalized over the brain?
It's generalized because it was shown that the brain was illuminated and the brain waste, for example, beta amyloid, was transported to the deep cervical node. So it's already generalized response, because from deep cervical nodes it will move to the lymphatic system and cleared from the brain. The same thing with blood at intracranial hemorrhage model. So the thing is, actually I can show you, no, I don't have the slides. There is a problem discussed in the public about penetration of the light. In terms of activation of meningeal lymphatic and brain lymphatic, it's necessary to penetrate through the layers of the meninges and a little bit in the cortex to activate the whole system. So in that case, the photobiomodulation is the perfect way to activate brain drainage system.
Okay, thank you. And also, I wondered if you had looked at photobiomodulation as a systemic treatment to photobiomodulation on the body, if it had any effect on the glymphatic system, or if it had to be transcranial.
Well, if I could enter into the body pad is what we put on the chest or the back of the neck, or any places for the mesenteric lymphatic dilation, which Denis referred to earlier. So you have a remote effect, but actually it's a direct effect to create that vasodilation within the meningeal and the mesenteric lymph vessels.
Yeah, it can be expanded to the full body, because we stimulate the brain lymphatics, by neck we stimulate cervical lymph nodes, and we can also stimulate whole body lymphatics to boost all these processes.
In the paper, we proposed that at least at this point, doing the light, immediately before bed, will improve the circulation and help with this pumping action. And then we would recommend that they do it in the morning also to create this drain. So it kind of works within the circadian balance of what the body's doing and what the light should be doing. That was kind of the conclusion of the paper.
Thank you. Yeah, I don't know if there's anyone else that wants to ask questions.
There is a question from Karen. She asked, has intracranial photobiomodulation have been done as a standalone treatment on humans yet? Have only seen studies using both at the same time. Yeah, I could not say that anything could. I saw this year there was a nice manuscript reporting photobiomodulation of traumatic brain injury in 3 patients with intranasal and head photobiomodulation, but I haven't seen about intranasal yet.
Can I just ask you, Denis?
Yes.
Thank you for your presentation. It seemed like a lot of that that you presented was an animal model, was mouse model.
Yes.
Is there a lot of human research to kind of back that up, or is it mainly being inferred from the anatomy of like the animal studies?
It haven't been done yet, but we plan to do it. Yeah, because it's complicated to monitor the outcome, because it's a little bit tricky. Then when animal, you can put it under microscope and you can do whatever you want, but it's in the plan. Maybe next time.
Mm-hmm.
Oh, there are some questions in the chat.
Yeah, so people who are putting questions in the chat, feel free to speak up, ask your question, or Denis, if you want to read them.
Can you speak to the outcomes measures to use which with humans to verify that photobiomodulation is activating the target system? It's a good question, and it have not been done yet, but technically it can be done probably with MRI, with magnetic resonance imaging. There is an attempt to do it with near-infrared spectroscopy, and with some indirect measures like heart rate variability, and in human it's much tough to see the activation of the meningeal or lymphatic system or glymphatic system, but potentially it can be done. Probably the best way is to directly show it is the MRI. And second question was, I am wondering what frequency of light was used, and if you found there to be a lot of differences between 660, 810, and 1072. We used a 1067, so it's a near infrared light. We haven't tried more short wavelengths.
Denis, mine was the question about the outcome measures. I just had a grant that went in through review, and it's hard to sell PBM to people who don't use it and who don't believe it. And even if this grant was well written, documented, I mean, one time I got feedback that said, everything that you said is clear, but it requires the reviewers to believe that this is a true effect. And so they're looking for some clear connection between the treatment and, the effect that you're positing is being produced. So as we know, many animal studies do not carry over into humans when, they're redone. So, I mean, you can demonstrate it in animals, but then it needs to be, reproduced in humans. So, that probably is the next step. but having outcome measures that are valid and serve as external criteria, I think are very important. So, you know, when I ask that question, I mean, maybe it's low hanging fruit, you know, to kind of say, yeah, what are those? Because that's true for any of them. But I think it's particularly important for PBM.
Well, I think, first of all, it depends on the pathology which you are testing, right? And was your grant on some disease, or it was a kind of safe study on healthy people?
Well, you know, one of them was on traumatic brain injury, and the latest one was on long haul COVID.
Oh, okay. Well,
I just want to say something. We're not conducting real-time fMRI and intranasal and transcranial stimulation of the brain. It's ongoing now. I have, you know, preliminary data, and I think this will be useful to answer the question, Jeff, that you brought up, and other people too.
Yeah.
Support a grant, because the brain response is actually very, very significant. I see, when I compare against transcranial magnetic stimulation and, you know, transcranial direct current stimulation, you know, looking at the literature, actually photobiomodulation, while it doesn't give you the feel of, you know, either tingling or some kind of a knock from TMS, actually it does show significant cerebral blood flow. And I can also say that the intranasal produces a lot of response from the brain too, at much lower power density. So, so take comfort. That is, maybe give us a few months. I think we'll show the results.
and
That's encouraging, though.
I think Denis could speak to the difficulties in human evaluation, and also our team member Nagesh, who's attending today. We're actively, you know, seeking the means to non-invasively demonstrate changes in the glymphatic system, but it is a, you know, we're on the cutting edge between technology and what you can do to humans. So we kind of to have, you know, valid measurements of the glymphatic system. Would you agree, Denis? Nagesh, if you want to chime in.
I agree. Yeah.
It just funded. Denis was just funded with a TBI case, and some of this stuff is going to be looked at.
Yeah, but it's in mice. Mice are easier. But I would say that it should include a very good battery of neurological outcome testing. Probably this is the best measure of outcome, because, okay, despite you might have increase in CBF or increase in something, if it does not show good outcome in cognitive and other things, it might be just not useful. Like there is a lot of batteries, and I hope at some point we'll do also the study, because I am working at Loveless Biomedical, which include Mind Research Network, where we'll do human studies, and we'll have dozens of different neurological testing for humans. And so far we were working on transcranial magnetic stimulation, transcranial direct current stimulation. I hope at some point I'll be able to do it on photobiomodulation too.
Sorry, can you repeat what type of testing you would be able to do?
Oh, we have Mind Research Network as a part of our organization, and this subdivision is mainly focused on human research, and we do a lot of neurological testing or cognitive testing for humans, and we combined with MRI and with magnetoencephalography. And so far, we have projects on transcranial direct current stimulation, transcranial magnetic stimulation, so in general, electrical or electromagnetic stimulation. So it's a question of funding, and I hope I will get some funds, maybe with in collaboration with somebody, and we'll be able to do that because we have technology, but no funding yet for that. And yeah, it's it's general problem for everybody, right? So
it will happen.
Yeah.
I had a related question. You mentioned brain ventricles pulsatility. Can you talk a little bit more about this physiological changes?
There is several good publication on when you see the pulsatility using MRI, and we even tried to. It's basically a functional magnetic resonance imaging. You need to use a specific algorithms to separate it, and we actually done it with MRI of patients during sleep, which was done in Siberia, and we repeated this manuscript. This described in the manuscript of Lutz experiment. And yeah, so this pulsatility depends on respiration and on heart rate. Where is it? Yeah. Oh, I didn't. Oh, there is a link to this manuscript of Fulls et al. In Science 2019. And I know they advanced in this direction much further in the analysis of CSF flow within the brain ventricles and subarachnoid space.
Yeah, Sarah and I had described this paper way back in the day when it kind of first came out, and that they found that the neurophysiological things like EEG, and then the heart rate, heartbeat, and then the CSF movement is when those things became sort of harmonized, it had a bigger push. That's one of the things that Denis is referring to. And, you know, what we're pointing out, and I think I had discussed with Paulo, is that what we're seeing in these people with the hydrocephalus or enlarged ventricle spaces is really a snapshot of a busted glymphatic system. You know, they've shown that the numbers like 60% of the brain, is shrunk during the sleep phase. Well, it's actually not cells that you're losing. It's actually a change in aquaporin giving up their water. And so they're trying to flush these things out. And that's why you have, this loss of, it looks like brain parenchyma and actually tissue, but probably this hydrocephalus or this swelling or increased intracranial pressure, if you will, which we're examining, would be one of the bell ringers of the early canary in the cave there, that you're not really draining the system. So there's blockages there. So, you can't really stimulate a toxic brain is kind of how we kind of say.
Yeah, and I forgot to mention the important thing. This brain pulsatility is really visible during the sleep phase. And it might be a good outcome measure for photobiomodulation, whether to increase this pulsatility. Because in awake state, it's even hard to discriminate it from other movements, and yeah, it's significantly increased during the sleep phase.
So how quickly does volume of the ventricle change, and how, so what's the magnitude of the change? I mean, besides the extreme cases. I mean, maybe there's a question for an MR researcher, but just wondering, and we can sort of look into the publication as well.
Nagesh might be able to answer that. He is the MRI.
Yeah, I think he'll be the best guy.
He's given us that figure before if he wants to chime in, but he's explained that before. There it is.
Cool.
Go ahead, Nagesh.
Yeah. Hi. Can you hear me?
Yes. Nagesh, we can hear you.
Hi. Yeah. I mean, I'm not sure about the exact magnitude or the percentage of the fluctuations that happens at the non-REM phase of the sleep, but that's usually like what we have been using as a, for instance, the CSF flow or the pulsatility at the cerebral aqueduct or the fourth ventricle. And what that paper has shown is that the CSF waves are like anti-correlated with the bold response. So it's like there is a reversal because during the slow wave sleep, you have reduction in the cerebral blood volume. And to compensate for this, there is the CSF flow which happens at these ventricular sites. And that kind of aids in flushing out these waste metabolites from the brain during deep sleep.
That's sort of what we're trying to examine, Paulo, with the Hanli-Lu data set. Is we want to see this, if it's a similar process, you know, activation, deactivation, what is the fluid, you know, what is the flow? And then those, you know, while it's difficult to see during those periods, maybe we'll be able to, you know, at least get the algorithm down to see these changes a little bit more accurately.
Yeah, and these physiological changes that Nagesh was alluding to kind of illustrate what would be the likely explanation of a greater effect of photobiomodulation during sleep if the brain is anyway flushed during that time more than other times. So it's a very interesting correlation. I know we have a few more minutes. I don't know if anyone else had other or different questions or topics.
I have a question, Paulo.
Go ahead.
In the case, Denis, in the case of hydrocephalus, so if the effect of photobiomodulation is related to the dissociation of nitric oxide, and nitric oxide is related to the vasodilatation, so you think maybe the condition can be like worsened by the photobiomodulation? So there's a side effect on this for this application, in this case of hydrocephalus or other case where the volume is already like higher?
Yeah, it's a good question, and especially it's complicated to answer because of various reasons for hydrocephalus. So it should be, maybe the best way is to use different animals models representing different pathological cascades of the hydrocephalus and to see how it works. Maybe actually a good project to compare different.
Denis is going to be doing some work with intracranial pressure measurements, you know, during, you know, after different times of the day.
Yeah, but it's a TBI, right? And it's TBI is a little bit different. Yeah, some part is to some part is as a sum of the hydrocephalus, but hydrocephalus is a very complicated topic. As in,
we're just looking at neural inflammation as it relates to increased intracranial pressure. That's one of the things that we're really investigating. Is it, you know, what is the correlates and what are the, you know, what are the different factors that you can look at for that also? Because, you know, if you bang your finger, it swells, right? If you bang your brain, it swells. So is there some correlation between, in the acute TBI model, this changes in pressure, and can photobiomodulation be effective on that?
Yeah, and I like to say that I don't show this data here, but intracranial hemorrhage cause a severe increase in intracranial pressure, and we've shown that photobiomodulation decrease it, probably due to increasing fluid outflow out of the brain cranium. And might be also another interesting topic, because like several years ago, I had a good project on intracranial pressure effect on cerebral circulation, where I have shown that when you increase intracranial pressure, it causes shunting of blood flow through the larger vessels that are not nutritive, and degradation of capillary flow. So, and it's interesting phenomenon, because when you use, for example, Doppler flow, you don't see the real changes, because some vessels are decreased, and capillaries are decreased, and some vessels which can be called shunt flow or maybe vascular heterogeneity increased flow. And when you average with Doppler flow, like in clinic, you don't see the difference. And you may say, oh, the cerebral blood flow is preserved despite the brain is starved. So, and I believe it will be a lot of offshoots from studying intracranial pressure increase and lymphatic, meningeal lymphatic and glymphatic system changes during this process.
I thank you for letting us go a little on our own. We missed our meeting this week, so we had a lot to chat about. Dennis, I think you did a great job educating, all of us, even, with the new stuff that's coming out, and highlighting the work of the Russians, who really, broke a lot of ground in this field. I appreciate your work.
Thank you.
Absolutely. Very, very cutting edge and very interesting work. We have maybe a couple of minutes if there are any other questions or quick questions.
Hello, this is Hanli Liu. Sorry I was late, but I tried to find the place where I can raise my hand. But anyhow, I just want to make a quick comment. This is a really interesting topic, and it's relatively pretty novel and new. And Joe had asked one of my students, former student, to work on this, but then he graduated. But now I have another new student that is learning and processing the data. So maybe we'll see. What we can do is really to see the correlation between HBO versus this water. So hopefully, maybe I'm thinking about another 2 months, we will be able to see whether we see a clear coupling or a correlation. If we do see it, then that would be a very good sign. Oh, by the way, just for the audience, what we do is really measuring frontal, prefrontal signals that covers the temporal changes of oxyhemoglobin and total blood volume and cytochrome C oxidase, oxidize the cytochrome C oxidase changes, and now we can have water included, and hopefully that will have some quantitative connections or correlation. So I'm just basically, since Joe mentioned that we're working on it, so I'm going to make effort to see whether we're getting some readings out of that. We have gotten 25 older adults measured under regular, what do I say, at resting state. But the data I'm talking about is really not photobiomodulation treated. But then we have another data set that have been including the photobiomodulation treatment. So there are, we just need time to get data processed. So it's sort of a pre-announcement about the study we're doing that. So hopefully by the end of the summer, we'll have something to show. That's it.
Let us know, Han-Li. We'd love to see it. I know that we are at the end of the hour. Thank you all, and as usual, to be continued. Take care.
Thank you.